I don't know, eventually. All right. So I asked you real quick to tell me some parts of the human body that are made of proteins, because this whole section is about copying DNA onto RNA, wiggling through it, going out into the, and finding a, to make a, by assembling, informing. So we're going to be dealing with that today, okay? I already got most of it in you already, I know. Yes, ma'am. I will be there at the end of the hour, I promise. Okay, keep them out. So today we'll look at gene expression, how we express genes in the form of proteins, the role that RNA plays in gene expression, and what happens during transcription. We'll look at things called codons and determine how they help determine the order of amino acid or the sequence of amino acids that occur during translation. We'll also look at the major steps of translation, the different components of reading mRNA to tRNA, codon to anticodon to amino acid sequence, and how we get traits that result from the expression of a single gene. What? Oh my goodness. I forgot to get your answer. I just started talking. So you told me muscles, cells, hair, skin. So my question is, could you put it just about any part of the body on here? Sternum, pectoralis. Head, shoulders, knees and toes, yeah. Fat would have, well, true just pure fat would have only adipose or fat cells, so that would not. So DNA, you guys get focused, provides the original information from which proteins are made in a cell, but DNA does not directly make proteins. Ribonucleic acid, or RNA, is the second type of nucleic acid, and this takes the information from DNA out into the cytoplasm, of course, to find a ribosome to make the protein. And gene expression, simply put, is the manifestation of genes into specific traits. So if I say gene expression, that means that I must have copied DNA onto, it must have wiggled through a, gone out into the, and found a, to make a, by assembling, and that's why we see the protein there, right? That would show up in the phenotype. So gene expression produces proteins by transcription and translation. That sounds like a test question, doesn't it? Like, what are the two parts of gene expression? Part 1, transcription. Part 2, translation, right? Bless you. So transcription is copying DNA onto RNA. Translation is when a protein assembles amino acids. Okay. So the first stage of gene expression is called transcription, and this is the process of making RNA from the information that is on DNA. So copying DNA onto RNA is called transcription. Transcribe. Are you guys copying notes right now? Yeah, that's a form of transcription. You're copying them down. So transcription is similar to copying or transcribing notes. DNA, my board up there, is kind of like the DNA, and you guys are copying it onto RNA. So you can take it with you, right? I can't fit the board through the door, but you can fit your notes through the door, right? Think of the door as a pore. It even rhymes. Listen here. Don't think about it too much. Right? Pretend it's a cube. What? I know. How about, I can't fit that wall through the door. All right, there. All right. Let's not be smart. Let's be dumb, right? That's what I used to say to my dad, and then he'd look at me like he was going to kill me. Don't be smart with me. I'd be like, what do you want me to be, dumb? And then Dad's head would turn red. Look at me like I was about to. Yeah. I don't remember much after that. I woke up. Might have been child abuse, but I don't remember. I was unconscious. So the second stage, lost my voice. The second stage of gene expression is what is called translation. This uses the information on RNA to make proteins by assembling amino acids and forming peptide bonds, right? So translation is similar to translating a sentence in one language, RNA, the nucleic acid language, to another language, the protein language or amino acid language. Think about it. There's two different macromolecules. First one is nucleic acid, DNA, and RNA. They're both nucleic acids, but then I somehow have to change it into a protein, which is made of amino acids, completely different things. So it's much like translating a sentence from one language into another. So you all understand that we copy DNA onto what type of RNA? Everybody say mRNA. That's called messenger RNA. So we copy DNA onto what kind of RNA? mRNA. mRNA wiggles through goes out into to find to make by assembling. What's the process called where we copy DNA onto RNA? What's the process called where we turn RNA into proteins? Translation. What are the two parts of gene expression? Transcription and translation. So all of the steps in gene expression are going to involve RNA. In cells, there's three basic types of RNA that complement DNA and actually translate the genetic code onto proteins. Now, like DNA, RNA is made of nucleotide subunits linked together. Remember that monomers of nucleic acids are nucleotides. The monomers of proteins are amino acids. So RNA differs from DNA in three basic ways. You need to know all three. First, RNA is composed of a single strand of nucleotides instead of two. So one major difference. RNA is single-stranded, DNA is double-stranded. Okay? Secondly, RNA nucleotides contain a 5-carbon sugar ribose. Remember we talked about DNA being named after deoxyribose? RNA is named after its sugar, ribose. Thirdly, RNA nucleotides have a nitrogenous base called uracil. Uh-oh. But they do not have thymine. So now, if I have an A, I have a, everybody say U. So in RNA, A does not go with T, A goes with U. And uracil is going to be complementary again to adenine instead of thymine. Let me explain that to you guys and kind of we'll talk about it a little bit. I'm going to give you guys a sequence. I'll start with a DNA sequence. Let's say I have a G, C. So we need to code this with an mRNA sequence. You guys should probably be writing this down so you understand. In an mRNA, since we're copying DNA onto RNA, A goes with U. So things change just a little bit. And G still goes with C, and C still goes with G. So the base pairing rule just shifted a little bit, didn't it? Okay. These three-letter words, see how there's three letters here? This is what is referred to as a codon. Codons, which we'll talk about, are these three-letter words that mRNA is read in. So we don't read mRNA U as one word, C as one word. We read UCG as one word, and then the next three would be another word. Okay? So that's one type of RNA. We copy DNA onto RNA. What type of RNA? Then mRNA wiggles through nuclear pore and goes out into the cytoplasm. Okay? And so I'm actually going to make a little barrier here just to represent the cytoplasm. Okay? So cytoplasm is this way. What's over here? Nucleus is this way, right? Dividing line. Say this is the borderland right here. Okay. All right. So when I go now to the next type of RNA, we'll talk more about the different types. We copy DNA onto RNA. What type of RNA? mRNA wiggles through and goes out into to find to assemble. Who carries amino acids? Everybody say tRNA. Transfer RNA. Say tRNA carries amino acids and has anticodons. Okay, so let's match them up. U goes with A, C goes with, and G goes with. These are called anticodons. So, we copy DNA onto what type of RNA? mRNA wiggles through everybody. mRNA wiggles through goes out into to find to assemble. By matching, everybody say tRNA, anticodons to codons to bring the correct amino acid into sequence. So that means it's a little more complicated than what we've been talking about, right? Yes? So it's a code. Now, I'll give you the decoder in a minute. You would never want to use the anticode, would you? You use the code. Where's the code? The code. Everybody say the code is on mRNA. And if this is how I remember. You know, it's also kind of a joke. You know why? You know what DNA says to mRNA whenever it leaves the nucleus? Don't forget to put your code on. So, but it helps you remember, right? So that's I don't get to tell that joke now. Now you got to use it as like a pegging mechanism. A deoxyribonucleic acid. Okay. So what is? Oh, yeah. Okay. So don't confuse anybody. So here's the thing, guys. You have to go from DNA to codons and mRNA to anticodons and tRNA. What else does tRNA carry? The amino acid. Now, we're going to get more into this, but if I ask you where the codons are at, you tell me mRNA. Okay? Sure is never a good thing to say to anybody, especially your boss someday, because they don't like to hear that. They like to hear yes or no. Yeah. No, don't ever. No's okay. If you don't get it, they want to know. Right? Yeah, sometimes. Sure is like a peeve word for me. It's okay. You didn't know that. But like, just to let you know, like adults in general hate the word sure. It means like, yeah, maybe I'll do that if I get around to it, you know, or maybe I kind of don't get it. Everybody hates sure, you know. Sure is like a deodorant that might work, you know. That's not good. You know. So the three main types of RNA in which play a role in gene expression are messenger RNA, transfer RNA, and oh my gosh, I just gave you another one, ribosomal RNA. The good news about, everybody say rRNA. Makes up the nucleolus. And then it makes up the ribosomes. Okay. So if I ask you what ribosomes are made of, you tell me rRNA and other proteins. So all you have to know for rRNA is it's what ribosomes are made of. Okay. So messenger RNA we talked about, it's produced when DNA is transcribed onto RNA, and the mRNA carries instructions for making a protein from a gene and delivers the instructions to the site of translation. And at the site of translation, tRNA, transfer RNA, reads the instructions carried by the mRNA and translates them to the mRNA sequence into protein subunits called amino acids. And then that rRNA, the ribosomal RNA, all you have to know is that that's part of the structure of ribosomes. We'll keep it very simple for that one. It's right in the name. What is rRNA? The stuff ribosomes are made of. So ribosomes are the cellular structure where protein production occurs. You guys know that. I've got that kind of drilled into your head. So let's talk. We copy DNA onto what type of RNA? mRNA. Everybody say mRNA. mRNA wiggles through goes out into to find to assemble by forming. mRNA has three-letter words called codons, which match with anticodons, which are found on, everybody say tRNA. tRNA, which also carries amino acids. Okay. We'll get there. So during transcription, the information in a specific region of DNA is called a gene. And this is transcribed or copied onto mRNA. We copy DNA one gene at a time. And transcription is carried out by a process called RNA polymerase. Oh no. We just talked about DNA polymerase yesterday, didn't we? Do not mix these up. It's very easy to do. DNA polymerase is involved when you're making more what? Everybody say DNA. DNA. RNA polymerase is involved when you're making RNA. Please keep those separate in your brain. It's very easy to mix replication and transcription up just because they have similar sounding things going on, including their enzymes. So transcription begins when RNA polymerase binds to one or the specific DNA sequence in the gene that is called the promoter. Everybody say promoter. So if I ask you where we start copying genes, you tell me the promoter. Promoter gets things started, right? Gets them going, like promotes an event. So, you ready? Okay. When RNA polymerase reads a promoter, it starts copying DNA onto let's do it again. When RNA polymerase reads a promoter, it starts copying DNA onto. When RNA polymerase reads DNA, it starts copying DNA onto an RNA strand when it reads a, now I'm messing with the promoter, right? I don't know if we're ready for this yet. We'll slow down. We'll slow down. You copy DNA onto, once you read a, everybody say promoter. Promoter. Okay. What type of RNA? mRNA. mRNA wiggles through goes out into to find a ribosome to make by assembling. What are the three-letter words found on mRNA? Codons. Codons match with anticodons, which are found on, which also carry amino acids. Okay? Right, I know. So RNA polymerase then unwinds and separates the two strands of the double helix to expose the DNA bases on each strand. Now, you guys, in DNA replication, it was DNA helicase that unzipped, right? And then it was DNA polymerase that copied the nucleotides. There is no helicase here. There's just one enzyme. How many enzymes? One. What's its name? RNA polymerase. Okay? There's just one. It does everything. So RNA polymerase moves along the bases in the DNA strand and adds complementary RNA bases as it reads the DNA of the gene. A's go with, not T's anymore, now they go with U's. G's with C's. A's with U's. T's with A's. A's are still there. Don't forget. Okay. So behind the moving RNA polymerase, the two strands of DNA close up and reform. And then they start to make that double helical structure, and it winds back up. You only unzip the parts of DNA you're going to read. Why? It has weak hydrogen bonds, and it would fall apart otherwise. Okay? So transcription is not the same process as replication. They're easily confused. They are easily confused. In transcription, a new molecule of RNA is made from the DNA, but in DNA replication, a whole new molecule of DNA is made from DNA. So here's the thing, guys. If I start talking about two enzymes, what process is that? No. If I said two enzymes, what process is that? I took that one off. What process is that? If I'm talking about two different enzymes, what process is that? We did it yesterday. DNA replication. If I talk about one enzyme, what process am I talking about? Transcription. Folks, there's two parts of making a protein, of gene expression. Transcription and translation. Transcription is when I copy DNA onto mRNA. I do this one nucleotide at a time. RNA is a little different. It's single-stranded. It has a different sugar called ribose, and it has uracil instead of thymine. So A's go with U's, G's still go with C's, T's still, when you copy from DNA and RNA, still go with A. A didn't disappear. It's just we don't have T when we make RNA after that. Okay? Once you copy DNA onto mRNA, it has these three-letter words called codons. Okay? mRNA wiggles through a nuclear pore, goes out in the cytoplasm, finds a ribosome. The ribosome is where mRNA and tRNA meet. mRNA has the codons, tRNA has the anticodons, which also carry an amino acid. That's how the amino acid gets in the correct order to form a peptide bond, so you can have a long polypeptide chain called a protein. Now, folks, that's more than all your parents learned in biology when they were in high school. I know that. You guys are expected to know more. Okay? There's a lot here. This is not a simple concept, I know. But it is the central dogma of biology. It is the rudimentary core of everything we've been talking about the whole year. Okay? So, the first part, copying DNA onto RNA, is called transcription. The second part out in the cytoplasm at the ribosome is called translation. Transcription, we copy the code. Translation, we decode the code, right? Okay. So when I look at transcription, you guys, there's one enzyme that does everything. It reads a promoter on DNA. Once the promoter is read, it unzips DNA, and it starts bringing in more nucleotides, making new hydrogen bonds, forming new side covalent bonds between sugar and phosphate groups, and makes a single-strand RNA. Here's another thing. Am I copying both sides of DNA or just one side? One. So in this process, I'm only copying one gene and one side of DNA. Can you read your book from right to left? No. So you have to read it from, don't be smart, you cannot. Okay? If you can, do it right now. You can. Okay. So, unless you read Chinese and we're in a Mandarin classroom, which we are not, you cannot do that. Okay. So now, if I look up there and I see I'm copying only one strand of DNA, everybody say template strand. That means one side of DNA is going to be the template, the other side we ignore because you have to read DNA from 5' to 3'. It's just like you reading your book from left to right. Okay? So you don't get to read DNA. Can I help you? Okay. Taking her out right in the middle of protein synthesis. Oh my goodness. All right. So you copy. You guys, let's shut the door. Thank you. Okay. So you copy DNA from 5' to 3', left to right. You don't get to read it backwards. So you can only copy one side of DNA at a time. So those three-letter words, what are the three-letter words called on mRNA? Codons. Everybody say codons. Codons. So mRNA has the codons. We're going to play a little bingo game maybe at the end of the day or tomorrow. But there's a 3-letter nucleotide sequence called a codon, and each codon is going to correspond to one of 20 different amino acids. So how many amino acids do I have? 20. How many codons can I make with 4 different nitrogenous bases and combinations of 3? 64. Okay? So codons act as start-or-stop signals for translation, and there are 64 mRNA codons. 64 codons and 20 amino acids. Do those numbers match up? So that means there's more than one codon for each amino acid. So sometimes there can be like 3 or 4, even 6 different combinations of G's, C's, A's, and U's that will make up that code for that specific amino acid. So this system of matching codons and amino acids is what is called the genetic code. You guys have heard of this before, right? The genetic code. It's like used everywhere. No one knows what it means, right? They just refer to it generally. This is why it's called the genetic code, because it's written in this secret language called codons on mRNA strands, which have to be translated by a ribosome through a process called translation and matching tRNA molecules that are carrying the right amino acid with their anticodon. So this is the chart that will make all of it seem a little bit more manageable. Would I ever want to read the anticode? Everybody say no. No. What do we read? The code. This will be the last thing we do today, but just listen. Okay? When you get an amino acid sequence, and I'll just write one on the board here, if I can find the pen I've left somewhere. There it is. Let me give you this. If this is what I copied from DNA, and this shows up at a ribosome, I have to be able to read this. What's the first letter in that? Hey guys, everybody do this together. What's the first letter up there? A. Okay, we'll come here. So we're in this row. First one's here. What's the second one? What's the third one? So if I come across where all these meet, what is that code for? Start. Now, start does code for an amino acid, and yes, you need to know this. It's called, oh man, it's called really messy writing. Okay. Methionine or methionine. Pronounce it how you will. I'm okay with saying either one. So methionine or methionine is the beginning of every amino acid on every single protein on planet Earth. This is a little creepy. It doesn't matter if you're a snail, a bacteria, a human being, a virus, doesn't matter what you are. You will all start every single one of your amino acids with AUG. Is that weird? Think about it. We all start with exactly the same amino acid in all 30,000 of our proteins, and it doesn't matter what you are on planet Earth. You will begin every one with AUG. You know that, you'd probably be a Nobel Prize winner, right? So, yeah, I wish I knew. I'd be rich, right? Really. So, no, I don't know why it does. And molecular scientists don't. That's just a fact that they study. Okay? So it's kind of like asking, why are we here? So, in a little way, it is, because this is the beginning of every protein that makes life. Okay? So here's the way I remember it. If you have to start work, you say what? UG. Okay, so it's like, okay, so you start with UG. Okay, always. AUG is the first codon you will read. Now, let's match this codon with its anticodon. What's the What's the anticodon of UG? A goes with U, U goes with A, and G goes with C. And this would be tRNA, right? And it would carry the amino acid methionine, whoops, also known as start. So if I ask you for the start codon, you must tell me that it is indeed methionine. And yes, all proteins start with the same one. Now, once Ms. Warner's done talking, I will give you a few more and we'll play around with it a little bit, but I'm going to stop here.